Thanks to the organizer for letting me present these interesting patient stories and also describe the utility of whole exome sequencing in detecting novel CDG genes. It started with one patient that was born 3 years ago at our hospital in Oslo, and she had a very specific phenotype and clinical presentation. She had SCID, severe combined immunodeficiency, and congenital neutropenia, and infections from birth with pneumonias and sepsis and gastroenteritis. In addition, she had a special skeletal phenotype. And what was interesting was that the radiologist at her hospital knew Gen Nishimura in Japan and contacted him and asked if he had seen something like it. And then he recalled that he had heard and seen X-ray pictures of a child with almost the same skeletal phenotype and also had the SCID disease. The third family, that's patient 2, was detected with exome sequencing because we did exome sequencing of all these affected patients. But this patient had the same SCID and congenital neutropenia phenotype, but didn't have the skeletal features. Let's go forward. I'm sorry, it's a little delayed here. Okay. As you can appreciate, both the patient 1 from Oslo, who had Afghan ancestors, and the German patient had almost the same facial features with mid-face hypoplasia, micrognathia, and downturned corners of mouth. They also have these thorax abnormalities with pectus excavation, and the skeletal features on X-rays were quite similar, almost as they were siblings. And here are the X-rays, and the radiologist emphasized that in our patient from Oslo, there was an extra ossification center, in addition to the exaggerated trochanter minor, which is on both proximal on both femurs, giving the appearance of a monkey wrench femur, and this was also seen in the German patient. The last patient, who was had parents from Mexico, had no dysmorphic features and normal development intellectually, and also had normal stature. So in summary, for all these, we found mutations in the same gene. They had eczema, abscesses, recurrent infections, which they shared, and this severe immunodeficiency with T and B cell deficiency. And the T cell deficiency made us give them the diagnosis SCID. They also had congenital neutropenia and progressed to bone marrow failure. Our patient in Oslo and the German patient had severe development delay and also the skeletal phenotype that resembled what we call Desebois dysplasia. So this is the immune system and how it develops from a hematopoietic stem cell into different subclasses. And what was special for this type of SCID in this patient is that it's not only T cell deficiency and B cell deficiency, but it's also combined with this neutropenia, and that hasn't been seen in SCID before. And 2 of our patients were treated with hematopoietic stem cell transplantation. Then we replace the blood progenitors, and 2 of them were cured, and the other one died at 7 months of an overwhelming infection just before the transplantation was planned. So what is a hematopoietic stem cell transplantation? We're not transplanting all stem cells, it's only the blood precursors. And it can either be done by extracting blood from the bone marrow of a donor, healthy donor, or from a umbilical cord blood. And the recipient, the patient, the sick patient, has to have preconditioning regimen with cytostatics to clear out all her own blood cells. And we're aiming for a healthy immune system, so when the recipient with an ordinary blood transfusion receives the hematopoietic stem cell from the donor, the cells will find their own way into the skeleton and start to produce different types of immune cells and blood cells. And we hope then to restore a normal, healthy immune system. So these are the 3 families. I'm not sure if you're used to this way of drawing family pedigrees, but the circles are females and the squares are male, boys or men. And we tested then with whole exome sequencing all these affected in the families, in the 3 families, and we found mutations in PGM3, phosphoglucose 3 in all of them. They had different mutations, and for some of them the pattern of inheritance didn't fit. For family 1, the family from Oslo, we found a homozygous mutation, and that was strange because the parents said that they were not related. But it turned out that they were from the same clan, so they're distantly related, very likely. And the small dots within the squares and circles, it means that both parents are carriers of this mutation. For the Mexican family, we also found a homozygous mutation. But that didn't fit because only the mom as you can see, only the mom has a dot in her circle. And that turned out to be, by using exome data, we could calculate and see that there was a deletion of the whole region on the paternal allele. So that's why it looked homozygous on our Sanger sequencing, but actually it was a deletion on the paternal allele, and the father turned out to be a carrier of this deletion, and he's healthy, but he's just a carrier of this deletion involving this PGM3 gene and a couple of other genes. And as you can appreciate, in this family, 2 other brothers had died of the same disease. We didn't know it by then, but they had both had neutropenia and overwhelming infections and died in their early childhood. Family 3 had a compound heterozygous mutation, and the parents were a carrier of 1 mutation each. So what could we say based on the exome sequencing? That we found mutation in PGM, and it may cause a novel glycosylation defect, PGM3 CDG. All 3 patients had 1 missense mutation, and it might direct us to that there should be a residual enzyme activity in these patients as well. And the protein structure of PGM shows that 2 of the missense mutations were in affected active site of this catalytic enzyme, and the other one bind substrate. So PGM is catalyzing the process in the it's towards the process of generating UDP GLUCNAC from glucose, and it's important for the catalytic activity of N-GLUCNAC 6P to GLUCNAC 1P. And it's UDP GLUCNAC is 1 out of 9 activated sugars, and it's important both for N-glucosylation and O-glucosylation. And it's also important for proteoglycan synthesis. So when you find a novel thing, what should we do? We should go to the model organism or other species and see if somebody has made a knockout mouse or something, so we can say and tell that the mice might have some of the same features. So what does the mouse say? The mouse, complete knockout, is embryonic lethal, but those with like hypomorphic mutation had 3 lineage cytopenias, just as our patients, and they were growth retarded. What was strange is that our patients had normal N-glucosylation, the standardized N-glucosylation, and O-glucosylation. But with using a mutant model with E. coli, we were able to demonstrate that the mutations we found had reduced enzyme activity. So, in summary, for all patients, we had eczema, abscesses, recurrent infections, severe immunodeficiency, neutropenia, and skeletal dysplasia. And one of the patients, the one with ancestor from Mexico, with a slightly milder phenotype, had also hyper-IgE. Almost at the same time as we published, 2 other groups published patients with milder phenotype or a different phenotype. It was 4 families published from Sassi et al. And 2 families from Sang et al. Altogether, 17 patients. And they had milder phenotype or different phenotype. Of course, they had infectious diseases. They had something with the T lymphocytes. And they also had, some of them had scoliosis. And the mutation, when you put them all together, they're spread all over the gene. But when we go and look at the function of these mutations, it seems like there is a genotype-phenotype correlation. And that reduced PGM activity caused reduced UDP GLUCNAC levels, which in turn caused reduced glycosylation branching. And other groups have studied this more extensively than we have. And it turns out that reduced UDP GLUCNAC levels cause reduced branching and a more severe phenotype, as has been reviewed in Yang et al. Recently. So, PGM3 is part, and PGM3 deficiency is part of, is filling up the puzzle in a lot of other defects in the glycosylation disorder puzzle. And for other diseases, like patients lacking MPI and PGM1, there has been trials with parallel treatment with mannose and galactose. And for us, for this PGM3 deficient patient, not for those that has been treated with hematopoietic stem cell transplantation, but for the other ones, there has now been discussed whether we could start with trial of with parallel treatment of other components, perhaps circumventing the PGM3 deficiency. And there is an ongoing trial that has started to recruit patients with PGM deficiency at NIH. So, how to diagnose those patients with CDG PGM3? They're not detectable on routine glycosylation tests. We can be aware of the various clinical features, but in the end, genetic testing and whole exome sequencing revealed the stories in all these 3 papers. But one important thing is that it might be detected on newborn screening. At least the patient with the most severe phenotypes with SCID. So those who lack T cells completely will or may be detected on newborn screening. So how come? This is because there's a new newborn screening that has been implemented almost statewide in US and are now about to be implemented in Europe as well. And then we're using the T cell, the immune cell, lymphocyte development to measure, quantify the amount of T cells generated. And how do we do this? When T cells are created, they cut out a small circular DNA, and we are able to measure that, and it's called TREX. So it's a circular DNA, can be measured in newborns on a blood sample from newborns, and if you have low or no TREX, you have a SCID. And it will detect PGM-CDG SCID. We have now retested our patient from Oslo, and yes, she didn't have, it wasn't 0, but she had low TREX, and she would have been detected on this newborn screening. So what happened with them? or patients after transplantation. Now it has been 1.5 year, 2 years since they were transplanted. There has been a progression of the skeletal dysplasia and the scoliosis in our patient from Oslo. But she is cured from her immunological problem, and she survived. Also, patient 2, who had bone marrow failure, survived and has no more infections, but he has also developed scoliosis. Regarding the brain and the cognitive deficit, our patient from Oslo had developmental delay from begin with, but there has been no progression of brain abnormalities on MRI scan. And the patient from Mexico, who also survived after the bone marrow transplantation, has had no progression or no development delay, only slightly speech delay in the beginning. Adult patients reported with the same disease with a slightly different phenotype were reported to have dysmelination on MRI, but those are adult patients. In conclusion, PGM3-CGG has a recognizable phenotype with immunodeficiency, skeletal features. It's actually not Descepois dysplasia. It's more called PGM3 immunoosseous dysplasia, even if all the other one causing Descepois dysplasia are also proteoglycan synthesis defects. There's a broad and variable phenotype. A genotype-phenotype correlation may exist, and it could be likely be related to the glycosylation branching. Routine screening for glycosylation gives you normal results. Only genetic testing reveals the diagnosis, and also track testing should be done and may reveal those who need the hematopoietic stem cell transplantation. Treatment. Hematopoietic stem cell transplantation cures the immune defect. It doesn't cure the skeletal dysplasia, and we don't know whether it will help with for the brain and the cognitive development of the children, and whether they will be prevented from this dysmelination observed in the older patients. Parallel treatment trials are ongoing and aiming to increase UDP GLUCNAC. I have a lot of people to thank, especially the people from Houston at the Baylor Hopkins Center for Mendelian Genomics, who did the exome sequencing in 2 of the families. And we also have collaborators all over the world submitting their patients from Germany and or radiologists from Tokyo, and my colleagues at Oslo University Hospital, who did much of the functional studies to prove that this was a really disease-causing mutations. And as last not least, thanks to the families. Thanks for letting me present these stories. Thank you.